Hello dear students, let's start to discuss one of the most important topic from unit human physiology for NEET UG examination. Now, commonly we used to say that it refers to the nervous system of our body, but why we use the term control and coordination over here we will discuss in this class, and which type of questions used to come in NEET UG examinations we will also discuss the same in this class. Dear students, the most fundamental characteristic of the living organism is that we are able to give response to the various environmental changes. So on the basis of this principle only we can identify whether an object is living or non-living. Now, living things are only able to give response to the various environmental changes. So we used to say that it is the fundamental characteristic of the living organism to give response to the various environmental changes, which are commonly named as stimuli. We used to give response to the change in our surrounding in the form of sound, in the form of temperature, in the form of touch, pressure. So all these changes are commonly named as stimuli, and we used to give appropriate response to these environmental changes. That is why we are considered as living only. So living organisms are able to give response to various environmental changes. Now the next question arises over here: does this response is achieved with the help of any single organ or any specific system in our body? Over here we used to say that during the generation of the response to specific stimuli, sometimes various organs or systems present in our body used to work in an integrated manner. The best example, if we are moving on the roadside and suddenly a dog start to run behind us. So in that condition, to save our life from the dog, we also start to run fast. To run the fast, our muscles need to work hard, and to provide the adequate energy to the muscles, our heart start to pump more blood towards the muscles, and inside the muscles aerobic as well as anaerobic respiration also starts, and the level of CO2 increases in our body. So we also start to do more forceful expirations to remove that CO2 from our body. So in this simple process in which we are saving our life from the dog, our muscular system, circulatory system, respiratory system, nervous system are working in an integrated manner. The process in which various organ and systems of a multicellular organism used to work in an integrated manner to generate an appropriate response is named as control and coordination. So the control and coordination refers to the process in which various organs and systems present inside our body works in an integrated manner to generate an appropriate response. We used to say in our body this response is achieved mainly with the help of two systems. These are nervous system and endocrine system. So in this chapter we will discuss in detail about the component of the nervous system and how the questions used to come in NEET UG examination. The first basic question may come from the component of the nervous system. So you need to remember the various component of nervous systems and small brief informations about them. We used to say our nervous system is mainly made up of 5 components. These are nerve cells or neurons, nerve fibers, supporting cells or neuroglia. These are supporting cells. Or neuroglia. Then ependymal cells and neurosecretory cell. So our nervous system is made up of the 5 major components. These are nerve cells or neurons, nerve fibers, supporting cells or neuroglia, ependymal cells and neurosecretory cells. Now, let's discuss about the structure of the neuron. Now, we used to say this neuron is mainly made up of 2 parts, cell body or soma and protoplasmic extension. It is the main part, cell body or soma, inside which nucleus is present, neurofibrils are present, mitochondria are present and nasal granules are present. We need to remember that these nasal granules are made up of rough endoplasmic reticulum. These are made up of rough endoplasmic reticulum and they helps in protein synthesis. They helps in protein synthesis. So, in the cell body, nucleus is present, neurofibrils are present, mitochondria are present and nasal granules are present. These granules are made up of the rough endoplasmic reticulum which helps in the protein synthesis. Now the protoplasm of this nerve cell used to come outward and these extensions are known as protoplasmic extensions, which are mainly divided into 2 parts on the basis of their size, dendrites and axons. All those protoplasmic extensions which are smaller in size are named as dendrite togetherly, whereas the longest protoplasmic extension is named as axon. The place where axon starts is named as axon hillock and where it used to get end is named as axon terminal. Now, inside the axon, neurofibrils and mitochondria are present. Inside the dendrites, nasal granules and neurofibrils are predominant. Now, on the basis of the structure of this axon, the nerve fibers are further considered of 2 types, myelinated nerve fibers and non-myelinated or unmyelinated nerve fibers. In myelinated nerve fibers, specific lipid-rich cell is present. Lipid-rich cell is present, named as Schwann cells, and these Schwann cells secretes myelin. They secrete myelin in peripheral nervous system. You need to remember, sometimes questions used to come that myelin is secreted inside the peripheral nervous system it is secreted by the Schwann cells. You also need to remember the functions of the nasal granules. Now, at few places this myelin sheet is absent and these regions are known as nodes of Ranvier. The conduction of impulse is faster. Conduction, need to remember, conduction of impulse is faster in myelinated neurons because charge used to get jumped from one node of Ranvier to another node of Ranvier, and this jumping conduction is known as saltatory conduction. Saltatory conduction. So conduction of impulse is faster in myelinated neurons. Now, so this is the discussion related with the structure. The next probability of question is refers to the classification of the neurons. We need to remember the classification of the neuron on the basis of their function and on the basis of their structure. So, if neuron is carrying the messages from sensory organs or sense organ to central nervous system, it is named as sensory. If they are carrying the messages from central nervous system to effector organs, named as motor neurons. If they can act as sensory as well as motor, they are named as mixed neuron. And if they used to analyze the informations, then they are known as interneurons. On the basis of their structure, these neurons are further considered of following types: apolar neuron, unipolar, bipolar, multipolar and pseudo-unipolar. Apolar, all the extensions are of similar in size, so we are unable to classify them into dendrite and axon, so named as apolar neuron. Such neurons are present inside the organisms of coelentrates. Unipolar, a specific one predominant protoplasmic extension is present. Such neurons are present in our embryonic stage. Bipolar, one dendrite and one axon is present, commonly find inside the retina of our eye. Need to remember this, bipolar neurons. They are also present inside the organ of corti and in retina of eye. The motor neurons of our body are multipolar in nature. They contain one axon and several dendrites, and pseudo-unipolar in which one extension used to give lateral terminal branches in our dorsal root. In dorsal root, such ganglions are present. So, first type of question, we need to remember the structure of neuron, about the cell body and the protoplasmic extensions, about the nasal granules, about the Schwann cells, and then the classification on the basis of function and on the basis of structure. Now, in nerve fibers, in myelinated, need to remember conduction of impulse is faster, conduction or transmission of impulse is faster inside them. Now, the supporting cells are further classified into two types: macroglia, larger in size, and microglia, smaller in size. These microglia are phagocytic in nature. They used to destroy the harmful pathogens. Macroglia is further considered of two types: astrocytes, star-shaped cells, provide physical support. Star-shaped, provide physical support. Oligodendrocyte, they secrete myelin in central nervous system. So, if the question comes myelin in peripheral nervous system, we need to remember that in peripheral nervous system, myelin is secreted by Schwann cells, but in case of central nervous system, it is secreted by oligodendrocyte. Now, ependymal cells, we know that inside our brain as well as in spinal cord, a fluid is present which is named as cerebrospinal fluid. Now, in the cavity of brain and in spinal cord, ciliated columnar epithelium is present which used to absorb the excess amount of cerebrospinal fluid, and this tissue is named as ependymal cell. So, ependymal cell is the ciliated columnar epithelium present inside the ventricle of brain and in the central canal of spinal cord, which used to absorb the excess cerebrospinal fluid. And neurosecretory cells are the endocrine cells which releases the chemical from their axon into the blood. So, we are discussing about the topic control and coordination, commonly named as nervous system. It is the fundamental property of the living organism to generate an appropriate response to the environmental changes named as stimuli. Now, control and coordination is a process in which inside the multicellular organisms, likewise in humans, various organs and systems of our body work in an integrated manner to generate an appropriate response for the stimuli. It is mainly achieved with the help of two systems, nervous system and endocrine system. The major component of nervous system are nerve cells, nerve fibers, supporting cells or neuroglia, ependymal cells and neurosecretory cells. Now, We had already discussed about the structure of the nerve cells and neurons and classification of the neurons on the basis of their function and on the basis of their structure. On the basis of function, sensory, motor, mixed and interneuron. On the basis of structure, apolar neuron, unipolar, bipolar, multipolar and pseudo-unipolar. Now, how these nerve cells used to work inside our body? The entire functioning mechanism based on two processes, the process of excitation and process of conduction. In the process of excitation, an electric impulse used to get generated on nerve cell in response to stimuli. So, whenever the environmental changes used to take place, the nearby nerve cell used to generate an electric impulse, or in simple form we used to say it used to generate charge on it. Now, two nerve cells never used to remain in direct contact with each other. The process in which one nerve cell transfers its impulse to another nerve cell is named as conduction. So, conduction is the process of transmission of nerve impulse from one nerve cell to another adjacent nerve cell. Now, we need to discuss how this excitation process used to take place and how this conduction process used to take place. So, we are discussing about the nervous system of human body. हमने component discuss करे, nerve cells, nerve fibers, supporting cells or neuroglia, ependymal cells and neurosecretory cell. And now we are discussing about the functioning mechanism. The functioning mechanism mainly based on two processes: excitation, in which in response to the environmental changes electric impulse or charge is formed, and conduction, in which this nerve cell transfer its impulse from one to another adjacent nerve cell. So that is how messages, if I will touch over here, then the nerve cell present in this region will first used to get excited, or it used to generate the electric impulse, and then this electric impulse used to get transmitted towards central nervous system. Now, the structures which helps in excitation are ion channels and sodium potassium pump. The active transport is achieved with the help of sodium potassium pump, and passive transport is achieved with the help of ion channels. What exactly they are? We know that the nerve cell contains the membrane on it, and this membrane is made up of the protein and lipids. There are small pores present in the protein particles of the membrane of nerve cell, which are named as ion channels, which helps in the passive transport of the ions. Means the ions will move from their higher concentration to lower concentration, and these ion channels are selectively permeable, means they will allow the passage of one type of ions only. Either they will allow the passage of sodium, potassium, calcium or chloride, They are further considered of two types: leak channels, which will always remain open; voltage gated channel, which generally remain closed but they open in response to stimuli. We will discuss further in detail. Now, these sodium potassium pump are electrogenic in nature. They used to work against the concentration gradient. Against means they will move the substances from their lower concentration to higher. Now, they used to causes the movement of 3 sodium from intracellular fluid to extracellular fluid and 2 potassium from extracellular fluid to intracellular fluid. Let's discuss this entire process in detail and understand the various stages of the excitations, which are polarization, depolarization, repolarization and hyperpolarization. Now, polarization, commonly named as resting stage of nerve cell. Resting stage of nerve cell. During this resting stage, this is the nerve cell, nerve membrane, and these are the leak channels. Leak channels are the examples of the ion channels in which substance are moving from their higher concentration to lower. So calcium is moving from intracellular to extracellular, whereas sodium is moving from extracellular fluid to intracellular. This is our intracellular fluid and this is our extracellular fluid. So sodium ions are moving from extracellular fluid to intracellular fluid and potassium ions are moving from intracellular fluid to extracellular fluid. The concentration of potassium is more. Potassium is 28 to 30 times higher, so they move more rapidly, whereas sodium is 10 times higher. In extracellular fluid. Over here, voltage gated channels are present, sodium voltage gated channel, then potassium voltage gated channel. These channels, voltage gated channel, remain closed. Now, then further there is a pump which is transferring 3 sodium on the outer side and 2 potassium on the inner side. This is sodium potassium pump. Now, so we are trying to understand the process of the excitation. There are different stages of the excitation. A nerve cell may remain in the resting stage, may remain in the active stage. Again, after active stage, it again comes back into the resting stage. So these stages are named as polarization, depolarization, repolarization and hyperpolarization respectively. Now, the first stage is polarization stage. During this stage, leak channels used to remain open. Sodium ions are moving from extracellular fluid to intracellular fluid. Potassium ions are moving from intracellular to extracellular. Which ion moves more rapidly or faster? Potassium, because their concentration is higher. Voltage gated channels remain closed, and sodium potassium pump. Now sodium is moving towards the inner side, तो अंदर accumulation equivalent ना हो जाए. For maintaining the concentration difference, this pump is transferring the sodium against the concentration gradient. यहाँ कम है, फिर भी पंप एनर्जी को यूज करके सोडियम को 3 सोडियम को बाहर ट्रांसफर कर रहा है और 2 पोटेशियम को अंदर ला रहा है. दिस इज सो कॉल्ड रेस्टिंग स्टेज. तो पोलराइजेशन को अगर हम डिस्कस करने जाएं तो वी विल से दैट इट इज नेम्ड एज रेस्टिंग स्टेज ऑफ द नर्व सेल in which leak channels remain open, voltage gated channels remain closed, and sodium potassium pump remain open. As a result, there will be more accumulation of the positive ions on the outer side of the membrane, and as a result, value of the resting potential becomes minus 32, minus 70 millivolt. Now, during this stage, we used to say that during this stage, there will be accumulation of more positive ion on the outer side of membrane and compared to it negative ion on the inner side of stage. This is known as polarization stage. Polarization stage. Now, immediately after it, the nerve cells used to undergoes into depolarization stage. During this process, the leak channels will remain open. Now, as soon as the stimuli or any environmental changes come near to the nerve cell, then this nerve cells open the sodium voltage gated channel and closes the sodium potassium pump. As a result, there will be a rapid influx of the positive ions inside the inner side of the nerve membrane, and it will lead to the generation of positive ion on the inner side of membrane and compared to it negative ion on the outer side of membrane. This is known as depolarization stage. Now, so in depolarization, we used to say that during it, in the presence of stimuli, nerve cells comes in the excitation, leak channels remain open, the sodium voltage gated channel opens, and sodium potassium pump used to get closed. As a result, more positive ions are present on the inner side of membrane, and the potential difference becomes plus 30 to 45 millivolt, which is named as action potential. So, polarization, resting stage. Depolarization, active stage in which nerve cell had generated the impulse on it. To generate the impulse, it used to open the sodium voltage gated channel and close the sodium potassium pump. So, during depolarization, it opens the sodium voltage gated channel and close the sodium potassium pump. As a result, charge on the inner side of membrane becomes positive and outer side it becomes negative, and the value of action potential becomes plus 30 millivolt. Immediately after the excitation, nerve cells transmit its impulse to another adjacent nerve cell and comes back in resting stage. So again, for Coming back to the resting stage, it will close the sodium voltage gated channel, open the potassium voltage gated channel and sodium potassium pump. So it again, repolarization means coming back into the resting stage after transmission of the impulse. It will close the sodium voltage gated channel, open the potassium voltage gated channel and pump. As a result, again there will be a more positive ion on the outer side and negative ion on the inner side. This is named as repolarization. And so repolarization, immediately after depolarization, nerve cells come back into resting stage. During this, leak channels remain open, sodium voltage gated channel used to get closed, potassium voltage gated channel opens and pump open, which will lead to the deposition of more positive ion on the outer side, and resting membrane potential difference again becomes minus 60 to 70 millivolt. That condition is known as repolarization. Hyperpolarization is the specific condition in which, in the presence of stimuli, instead of opening of sodium voltage gated channel, potassium voltage gated channels used to get open, which will lead to the deposition of more positive ions on outer side, and the resting membrane potential becomes minus 90 millivolt. That is known as hyperpolarization. So the best question whenever used to comes from the nervous system, it used to come related with the various stages of excitation. So many types of question can come. In polarization, what is the stage of sodium voltage gated channel? They used to remain closed. What is the charge on the inner side of membrane? Negative. What is the value of the potential difference? Minus 30 to 60. What is the major cause of the excitation? That is the movement of sodium ion due to the opening of sodium voltage gated channel. As a result, there will be a rapid influx of sodium from the extracellular fluid to intracellular fluid. So rapid influx of the sodium will lead to the depolarization due to the opening of sodium voltage gated channel and closing of sodium potassium pump. More charge on positive charge on the inner side of membrane, the value of action potential becomes plus 30 to plus 45 millivolt. So these are the different stages of excitation. Now the functioning mechanism depends on two processes, excitation and conduction. The next part is conduction. Conduction, it is the process in which nerve cell will transfer its impulse to the another adjacent nerve cell, and it is achieved with the help of nerve synapse. Synapse are the small gap present in between the two adjacent nerve cells, with the help of which the impulse used to get transmitted. Now, on the basis of the size of these synapses, we used to classify them into two parts: electrical synapse and chemical synapse. Impulse transmission is faster in electrical synapse. Impulse transmission is faster in electrical synapse. The gap is 0.2 nanometer only. So impulse can directly transfer from presynaptic membrane to postsynaptic membrane. Such synapses are found inside the smooth muscle fibers of the intestine, cardiac muscle fibers and epithelial cells of lens. Now, the most common type of synapses are chemical synapses, in which gap is up to 20 nanometer. In the structure of them, the presynaptic membrane is present, synaptic cleft and postsynaptic membrane is present. This is the part of the axon terminal. The nerve cell which is excited, उसका axon terminal part है. ये next dendrite का surface है, which is named as postsynaptic membrane. एक्सोडेंड्राइट सिनेप्स को हम डिस्कस कर रहे हैं. नाउ ड्यूरिंग दीज केमिकल सिनेप्स the impulse transmission is achieved with the help of chemical substances named as neurotransmitters. Now, how this used to take place? Now, the first step, impulse changes the permeability of presynaptic membrane and allows the movement of calcium ions. So calcium ions start to move from synaptic cleft into presynaptic membrane. Impulse जैसे ही यहाँ आएगा, permeability presynaptic membrane की change हो जाती है. Calcium ions moves inside the presynaptic membrane. It lead to release of neurotransmitter from synaptic vesicles into synaptic cleft. So now it causes the rupture of the synaptic vesicles. As a result, these neurotransmitters are released. These neurotransmitters are released into synaptic cleft, and now these neurotransmitters attached with postsynaptic membrane and causes depolarization. तो दो न्यूरॉन्स थे, फर्स्ट पर इम्पल्स पहुंचा, इम्पल्स पहुंचते ही प्री सिनेप्टिक मेम्ब्रेन की परमेबिलिटी चेंज हुई, कैल्शियम अंदर एंटर हुआ, कैल्शियम ने सिनेप्टिकल वेजिकल को ब्रेक डाउन करा, रप्चर करा, रप्चर करते ही न्यूरोट्रांसमीटर रिलीज हुआ, न्यूरोट्रांसमीटर पोस्ट सिनेप्टिक मेम्ब्रेन से अटैच्ड हुआ और उसको डीपोलराइज कर देगा. दैट इज हाउ इम्पल्स यूज्ड टू गेट ट्रांसमिटेड. Now, on the basis of their characteristics, these neurotransmitters are further considered of following types. Excitatory, they increases the speed of impulse transmission. Example of such neurotransmitters are acetylcholine, epinephrine, norepinephrine, serotonin and 5-hydroxytryptamine. Inhibitory, which decreases the speed of impulse transmission, gamma aminobutyric acid, glycine and mix, which can increase or decrease. Examples, dopamine. So till now we had discussed what exactly the term control and coordination is. Process in which various organs and systems of our body will use to give appropriate response to the various environmental changes. Now it is achieved, this response is achieved with the help of nervous system and endocrine system. The component of nervous systems are nerve cells, nerve fibers, neuroglia, ependymal and neurosecretory cells. Now the classification of the neuron on the basis of their functions, on the basis of their structures, need to remember questions used to come from these classifications and on nasal granules and Schwann cells. Then the functioning mechanism of nerve cells, process of excitation and process of conduction, the different stages of the excitation, polarization, depolarization, repolarization and hyperpolarization, and after that the conduction of the nerve impulses with the help of synapse, electrical synapse and chemical synapse. It's over for today. In next class we will discuss about the structure of human nervous system.